Good afternoon, everyone. Welcome to our SynGen series webinar. My name is Dan, and on behalf of Oxford Global, I would like to thank you all for joining us today. Today, we will have Guilhem Tourniaire, Scientific and Managing Director at Cellenion, who will be presenting on optimizing circulating tumour cells isolation for scRNA sequencing analyses. After the presentation, we will have a 10 minutes question and answer session. Please submit your questions through the chat option on the bottom of your menu display. The full recording of the webinar will be made available to download on our secured website, and the details will be sent to you after the webinar. So without any more delays, I would like to get started with Guilhem's presentation. Guilhem, over to you.
Fantastic. Thank you very much for the introduction. So good morning, everybody. So today I'm going to present you the work that we've been doing in collaboration with the hospital here in Lyon, the group of Professor Léa Payen, in order to isolate single circulating tumor cells for single-cell RNA-Seq analysis. But before starting this, I would like to do a small introduction about our cellenONE technology, and I will take you through a few examples of application of our technology developed by our customers, in particular in the field of transcriptomics, but also in the field of genomics and proteomics. And then I will move on to the main topic of this webinar, which will be the work that we've done to optimize the protocol for single circulating tumor cell isolation. So first of all, why do you need to isolate single cell? Well, probably most of you who are listening to this don't need an introduction to this, but with our system, we address two main applications: the field of single cell analysis, where you need to isolate your cell before analyzing them. And this is widely used for the study of the heterogeneity of tumor in order to identify certain subpopulation or better understand the mechanism involved in the cancer development. On the other hand, our systems are also used for cell line development whenever you need to do automated cloning. And this is more addressing the field of stem cell, monoclonal antibody development, or people undertaking CRISPR-Cas9 modification. But this will not be presented today in this presentation. I will focus on application in sample preparation for single-cell analysis and all the omics field. So our technology is based on a not so new actually technology, which was developed by our mother company Cellenion about 20 years ago. It is a piezoacoustic based technology, and it consists of a glass capillary, which is surrounded by a piezoceramic. And whenever we're going to send an electrical pulse to the ceramic, this is going to generate an acoustic wave inside the glass capillary, which will then eject a small droplet. So this technology can be used to generate very small droplets from 50 to 800 picoliter per drop. And we can obviously use this technology for single cell deposition, which is what we do at Cellenion. But you can also use the technology for dispensing nanoliter volume or picoliter volume of reagents. We have this little animation that's going to explain you the principle of the cellenONE technology, which is a single cell dispenser, addressing, as I said, the two fields of single cell analysis and automated cloning. The device looks like this. Inside the device, we have a holder for two MTB plates. These plates can be 96 well plates, but also 384 or even 1536 well plates. And we are also working with some micro wells, so structure which are very tiny wells. We control the temperature of these plates in order to prevent RNA degradation and also to improve the viability post-cloning. It all starts from a cell suspension. We aspirate the cell suspension into the dispenser. And then once per day, you're going to have to do a mapping step. And this mapping step is going to consist of the tracking of the cells throughout the nozzle. And from this, we can deduce the size of our ejection zone. And this ejection zone has more or less the volume of one droplet. And basically, we now have an algorithm that's going to detect the cells throughout the nozzle. And whenever we have a single cell detected in this ejection zone, we know that in the next droplet that we will generate, these cells will be ejected together with the droplets. So now we are looking for events. Every time there is an event with a single cell in the ejection zone, we know that the next droplet will contain a single cell. We move to the target. We send the droplet containing the single cell. If we have empty droplets, we discard those droplets. If we have droplets containing more than one cells, we also discard those. But when I say discarding, actually we do that into a recovery tube. And you can, after your first experiment, if you are working with very rare cells which you want to reprocess, you have the ability to collect those cells again. We do a number of measurements on the cell. We measure the diameter, the elongation, and the circularity. And this is used to differentiate a single cell from agglomerated cells, which would have a very different elongation and circularity factor. So this allows us to ensure that we only isolate single cell. And we do this until all the positions that have been set in your microplate or in your target are filled. In terms of throughput, we are now working at a throughput of about 100 single cell isolated in 2.5 minutes as we have improved our system. So in summary, the technology provides you with a very high accuracy. It is also provide a very high viability, but I will not describe this, and it allows you to inspect your sample. You can see if you have debris and all those other things. So this is nice, obviously, it's a nice animation. You can get an idea of the principle of the system, but you probably are wondering what it looked like in real life. So we have this little video now, which is going to show you what happened in real life. So on the left, you see the inside of the equipment. In this example, we are dispensing single cell onto a microscope slide that has a microstructure on top, microwells. And on the right, you will see the screen of our software. Actually, the old version, we have made the software a bit more sexy nowadays. But essentially what you have to look to is, so what you see here is the tip of the dispenser. The cells are going to travel from the right to the left of the dispenser. And the ejection zone is the zone from the left of this image to the first red line. This is our ejection zone. So whenever we're going to have a single cell in this zone, you will see on the left the dispenser moving, the axis moving to the target, generating the droplet, going back to the camera and looking again for a single cell event. So I'm now going to start the video. So a single cell in the ejection, we dispense. Single cell event, again, single cell event. We have a doublet. We don't want that. We discard it. No movement of the head. Again, some more single cell coming through. As you can see, it's a very heterogeneous sample of prostate cancer cells. Again, a single cell. We are also able to discard debris. You will see this in an example soon. So here is the debris. We don't want to isolate this, so we just discard it. And we do this until all the positions have been filled. Move on. So basically, the technology you just saw, we trademarked it under IBSCI, which stands for image-based single-cell isolation. It is kind of the equivalent of a bright field microscope, if you want. So you get a black and white image. And this technology was developed now over 2 years ago. But working with our customer, we had a number of requests after we first released this technology for implementation of fluorescent sorting, the ability to be able to sort certain subpopulation. And to do that, we implemented a fluorescent module into the system. And this technology is called FIBSCI, for fluorescent image-based single-cell isolation. And it can be used with up to 4 excitation channels. So, and what are the advantages of using this IBSCI or FIBSCI? Well, the advantage of IBSCI is that it is faster as there are less images generated and analyzed. It is a completely label-free technology. You get a high-resolution image for your isolated cells, and you can only do sorting by size or morphology, which is kind of limited depending on your application. On the other side, with the FIBSCI technology, you are able to do isolation of only live cells. If you use staining using a live dead kit, for example, you can sort certain subpopulation if you only want to isolate certain cells of interest. In the example that is of interest to us today, I will demonstrate to you how it can be used to isolate very rare cell population. And in the field of cloning, it can be used for isolation of the best expressors, for example, by sorting the highest fluorescent intensity of GFP, for example. And we are also developing some new application in the field of bacteria isolation, which was challenging due to the size of the bacteria, but now with fluorescent marker, we are able to achieve very good results for single bacteria isolation as well. And obviously, those two technologies can be combined, so you can sort by fluorescence, but still get a high resolution image after of your cells before isolation. So now I'm going to give you a few examples. in different fields of omics. The first example will be a transcriptomic example developed at the University of San Francisco. There, they are working with very rare cells, which is kind of relevant to our topic today as well. In this group, in the group of Michael Wilson at the Vail Institute in the University of San Francisco, they are studying samples of cerebrospinal fluids taken from a spinal tap from multiple sclerosis patient, and they wanted to isolate lymphocytes present in this cerebrospinal fluid. But the main challenge in this experiment is that the sample contained very, very few cells. The procedure is very painful for the patients. So the first thing the group of Michael Wilson wanted us to demonstrate was that we were not going to lose too many cells during the process of isolation. So we did a proof of concept study. In this proof of concept study, we prepared PBMC samples, about 30 microliter of sample containing about 450 cells per sample, which is the equivalent of about 15 cells per microliter. We took 5 aliquots of 5 microliter from this sample, put this into microwell plates, analyzed under microscopy to count exactly how many cells were present in this sample. And then we took 1 aliquot of 5 microliter, put it through the cellenONE system, and asked our system to isolate as many cells as it could. And the results with the first sample was that, on average, the sample contained 68 cells, so very few cells. Using the cellenONE, we were able to isolate successfully 64 single cells. So we repeated this process with another 2 samples, and on average, we obtain single cell isolation results of 95% recovery, which is unseen. We don't know any other automated system that can achieve such a high recovery rate. So this demonstrates that the technology is really suited for isolation, not only of many cells, but also very rare population of cells. Collaborating with another group at UCSF, at the CAT Center, the group of Eric Shaw, and this is some work done by Derek Bogdanov. He developed a method to do RNA-Seq libraries. So this is the method. So he used our system for doing single cell isolation, but then he used the nanoliter dispensing capabilities to generate libraries in nanovolumes. And what you are seeing here is a chip that they've developed there, which contain already a barcode and a single cell. And now they are doing the addition of the lysis buffer and the RT mix, 40 nanoliter in each well. By doing this, they are able to drastically reduce the cost of the preparation of your RNA-Seq libraries down to a cost of about 7 cents of a dollar per each single cell analyzed. And if you look at the price that other technology typically costs, you can clearly see the benefit of using our technology not only for isolation, but also for library preparation. I will now move on to a second example. This is some very nice work done at the University of British Columbia in Vancouver and the British Columbia Cancer Research Center by the group of Sam Aparicio and Robin Koop. They recently published a very nice article on bioRxiv, and this article actually has been just accepted in Cell and will soon be published in that very nice journal. And the method they've developed is for whole genome sequencing, and it is called Direct Library Preparation Plus. Some short words about this method. In their studies, they were able to sequence over 40,000 different single cells, and using their technology, they are able to identify stochastic aneuploidies, genome replication states, and clonal repertoire. So they are ideally suited to study the heterogeneity of patient tumor. And to date, so this was published just over a year ago in bioRxiv, and to date they've analyzed over 400,000 cells from patient samples in that British Columbia Cancer Research Center. And this technology is again based on a microwell chip, which contains about 5,000 wells in which they do the deposition of single cell. They verify the accuracy of deposition using a fluorescent imaging technique. Then they do the addition of the lysis buffer, heat lysis, and then add the reagent to prepare the library. But what is very unique about this technology is that there is no preamplification. So as a result, there is very little bias in the results obtained, and you obtain very high quality libraries. They are able to use a wide range of cells, so they've demonstrated applicability of this technology with nuclei and cells that were up to 80 micron in diameter. The technology is it's an open technology. It can be adapted into any other laboratories throughout the world, and actually a number of other cancer research centers are now adopting this technology, and it can process thousands of cells in each experiment. Again, they did some calculation in terms of cost of reagents for the preparation of those libraries, and they demonstrated that it costed them about 0.3 cents, 30 cents of a dollar for each single cell. So again, very interesting economically. Finally, I will give you a last example of a field where we see lots of traction at the moment, which is the field of single-cell proteomics. And I will present you an example of work that has been published over the summer in analytical chemistry and that has been done at the BAM Research Center in Berlin. In this work, they used a technology which is called laser ablation ICP-TOF-MS to study a number of protein and metal inside single THP1 cells. So in the first part of this paper, they describe how they used metal-based molecule in order to detect the protein content of cells and also the DNA content of single cells. And in the later part of this paper, they demonstrated that they were able to measure also endogenous metal quantitatively into single cell. And the last diagram on the bottom right shows you the change of phosphorus amount into the single cell as the cell goes through their cell cycle. So this demonstrated that their laser ablation ICP-TOF-MS technology allowed quantification of both protein and metalome studies at the single cell level. And we are working with a group of several other groups in this field, and we hope that some more paper will be coming out soon. So now I will move on to the main topic of today's webinar, which is the isolation of circulating tumor cells, which has been done in collaboration with the Institute of Cancerology here in Lyon and the team of the CIRCAN team of Professor Léa Payen. Most of the work has been directed by Jessica Garcia and François Monjaret from my team. And this was done in collaboration with a company called Biolytics from Singapore, which has a very nice technology for CTC enrichment. But first of all, I would like to tell you a few things about the importance of studying circulating tumor cells. As you know, you might know, circulating tumor cells are an integral part of the cancer disease. They are considered to be the seeds of the metastasis, so they are a very good indicator of metastatic changes. And you probably all know that in cancer patients, typically the primary tumor is not the killer, but the metastasis is. And nowadays, as you know, the cancer diseases are very complex and heterogeneous and dynamic, so it's very important that clinicians are provided with new real-time, minimally invasive and highly repeatable and comprehensive monitoring tool. And we hope that the use of these circulating tumor cells will answer this need. Nowadays, CTC are used in a number of different contexts. They can be used to screen for cancer by identifying the presence of CTC in blood. You can evaluate the severity of the cancer by counting the number of CTCs that are present. You can follow the trend in the CTC number, which can give you an idea about the effectiveness of a treatment. But here, what we want to really capitalize on is the personalized medicine and the field of ultra-precision medicine, what they call, which by being able to analyze the transcriptomic content of each of the single CTC, we are able to provide better personalized cancer treatment to the patient and also monitor those treatments over time. So why is it so challenging to study CTC? It's all down to numbers, actually. Just to give you an idea, in blood, you have about 1 CTC for every 10 billion of cells. So they are very, very, very rare. And what makes it even more difficult is that those CTCs tend to be very heterogeneous. not only in size, but also in the marker they are expressing. Nowadays, today's on the market, there are many different technologies for enrichment of CTCs, and these are based on two main principles. Some of them use physical properties. They isolate the CTCs based on their size or stiffness or density. But the main disadvantage of those technologies is that typically the smaller CTC will be missed. On the other hand, you have technology based on immunocapture with antibody linked to, for example, magnetic particles and other principles. These use typically the EpCAM marker or the cytokeratin marker. But the problem of this approach is that not all the CTC are expressing the same, those epithelial markers. And especially it is well known nowadays that CTC undertake EMT transition and will undergo display more mesenchymal phenotype sometime. And these are actually the more aggressive CTCs, and they will typically be missed by those technology. To give you an idea of the results of those enrichment technology, typically they achieve a log5 to log7 enrichment. So what you end up with is not a pure CTC sample. It's actually a sample that contains about 1 CTC for every 1,000 cells, and typically those remaining cells are going to be white blood cells. In our study, we collaborated with the company Biolidics from Singapore, which has developed the ClearCell-FX system. It is a label-free enrichment technology, which is based on the DIN fractionation principle, which uses a microfluidic chip to isolate circulating tumor cells. It is a very nice and automated system. It is relatively fast. And more importantly, it provides you with intact cells. So here you can see the white blood cell going down to the waste channel, whereas all the CTC are moving to the top channel for recovery and downstream analysis. So this is a protocol we have developed. It consists of, we start from a blood sample obtained from patients or prepared at the CERCAN Institute. We do the enrichment using the ClearCell-FX1. We obtain an enriched CTC sample. Then we take this sample, put it through our cellenONE system in order to isolate only the CTC. From the isolated CTC in a 96-well plate, we then prepare single CTC libraries using New England Biolabs NEB-NEXT single-cell kit. Once the library are prepared, we sequence them using a Illumina sequencer, and then we do the bioinformatic analysis. But what is challenging with this protocol is always those numbers. As I told you before, we start from about 1 CTC in 10 billion cells. After enrichment with the Biolidic system, we will obtain about 1 CTC in 1,000 to 10,000 white blood cells. And this concentration is typically too low, or there are too few cells of interest for other technologies that are used for single-cell sequencing, such as the 10x genomics technology or FACS. So we believe that our cellenONE technology, given the high recovery rate that it can achieve, was a very good proposition to achieve this. In this experiment, as everybody do in the field of CTC, we always start with spiked sample of known number of cells. And we used a very nice model developed by a researcher here in Lyon, which are those human mammary epithelial cell, HMEC, as CTC mimic, because those HMEC cells can display the epithelial mesenchymal transition. So they are very heterogeneous in their phenotype. And so they are very good model for CTCs. So, as I told you at the beginning, we have a number of options using the cellenONE that we could use to try to isolate those single CTCs. The first option was to use just IBSCI and sort by size. The advantage of this is that, as I said, it's faster than using FIBSCI, but the main disadvantage is that we would be missing most of the smaller CTCs. The other option is to use fluorescent-based isolation. First option, to use positive sorting using EpCAM or cytokeratin or both markers. This would be faster than negative sorting, but we would be missing all the cells that have undertaken the EMT transition. And these, as I said, are the most relevant cells that we want to isolate. So we decided to go against this option, and we opted for a negative sorting. So labeling all the white blood cells that are present in the sample and asking the cellenONE system to isolate only the non-fluorescent cells. This, in theory, could achieve much higher recovery rates of the CTC, but would be a bit slower than the positive sorting. And the main risk of this option was coming from the fact that maybe not all the white blood cell would be labeled with a CD45. But we went for this second option, and we will still use the IBSCI at the end to verify that only single cells are dispensed in our droplets. So we did the preparation of some mock sample. We started with 10,000 white blood cells, stained with CD45 only in the first experiment. We added 100 HMEC cells in this sample, and we processed it with the cellenONE. What we noticed is that we only recovered about 37% of the HMECs that were present in this first experiment. And when we analyze further the sample, what we realize is that about 6% of the white blood cell were not labeled. So the CD45 labeling alone was not enough. So we repeated this experiment, but this time by doing a dual labeling with CD45 and CD15 using the same fluorophore. And there we were able, with a 1% HMEC concentration, to recover 87% of the CTCs present. And we repeated also this experiment at even a lower concentration of 0.1%, where we demonstrated 100% of CTC recovery. So we were really happy with these results. From this, so this is our optimized protocol. So we now start from the spiked blood sample or patient sample, do the enrichment with the biolytic system, do a concentration step and the immunostaining using both CD45 and CD15, isolate the single cell into individual worlds, prepare the library, sequence, and do the analysis. And we got some initial results of analyzing those HMEC cells. And our libraries clearly demonstrated that this HMEC was showing a very high heterogeneity. And most of the cells, so we prepared library for 20 single cells, and most of those cells, 18 out of 20, were displaying an intermediate profile. And the single cell 17 was, on the other side, was displaying a clear mesenchymal profile, and the number 12 was still in the epithelial profile. Sorry about that. So this clearly demonstrates that our process is able to isolate single circulating tumor cells and prepare the library from those. So in conclusion, the combination of the ClearCell-FX1 system and the cellenONE F1.4 has been demonstrated using a dual immunostaining CD45, CD15, and negative selection is very promising for achieving high single cell CTCs recovery. We can prepare good quality libraries using the NEB NextSeq kit, and we have a number of additional work that will be done prior to the publication of this research work. So the next step will be to try to implement a freezing step after the enrichment with the biolytic system, and this will facilitate the logistics, because as you can imagine, it's a fairly long process, and the ability to freeze those samples will facilitate the logistics of this process, and also being able to process the sample for isolation in different sites, for example, different locations. After that, the Professor Léa Payen will process some lung cancer patients, non-small cell lung cancer patients, and prepare single cell RNA-seq libraries from those. And obviously, we would like to apply this method to other type of cancer to demonstrate that this protocol can be used for a wider range of patients. So in conclusion, I would like to acknowledge our collaborator, especially the team of Professor Léa Payen and Jessica Garcia, who has been instrumental in doing most of this work, and my colleague François Manjaret, who has done most of the isolation with our cellenONE system, and also Jean-Philippe Aurel from Biolidics, who has been a very good help to develop this collaboration, and Nicolas Doll at New England Biolab in France, who has been supporting this work as well. Finally, just a few words about our partners and reference sites. Presented here today the work from the group of Michael Wilson and Eric Shaw at UCSF, and also the work on whole genome sequencing done by the group of Professor Sam Aparicio and Robin Koop at UBC and the BCCRC. I would like to thank you for your attention, and I will be pleased to answer some of your questions. Thank you very much.
Thanks, Guilhem, for this presentation. Now I'd like to open to the floor for discussion. So remember to send us your questions using the chat feature of the platform. So first question, have you looked if the CTC enrichment step and single CTC isolation had any effect on the cell's transcriptome?
So actually it's a very good question. It's one of the first experiments that was carried out. We did library preparation on bulk and single cells at the different steps before enrichment, after enrichment, and after single cell isolation. And we saw very little effects of the different system on the transcriptome. So we believe that there is no major effect of our protocol on those cells' transcriptome.
Okay, thank you. And I'll go into this question, which leads on from that one. So can you explain again what exactly is enriched CTC and what enriched exactly means?
Well, it's enriched from a blood sample. So the blood sample will contain all the red blood cells, the white blood cells, and very few CTCs. After enrichment, an enriched sample will be a sample containing a higher proportion of CTC versus white blood cell, but these are never pure samples. So as I said in my example, using the Biolidic system, you end up with about 1 CTC for 1,000 white blood cells.
Okay, and what is the size range of CTCs that can be captured?
It depends on the method that you are using. In our work, we basically captured any CTCs that would be larger than 10 microns.
And how do you make the dead or live cells if one channel with propodium iodide isn't sensitive enough?
Well, so that's not related to the CTC work anymore. It's more related to the FIBSCI. How do we use the FIBSCI? I would just use something else than propodium iodide maybe. But so typically we have done some work with like cell tracker and propodium iodide, for example, and these typically are not a problem. You will detect live cells using the green channel, so the FITC channel typically, and then we would verify if those green cells are also orange, associated with the propodium iodide. And if the cells is only green, we will isolate it. If the cell is green and orange, then we will discard it. That's just like setting the parameters in our cellenONE software. Fairly straightforward.
Okay, and for the two techniques you show us, what about the index of the library? When do they dispense them before pooling?
So typically in those two examples, so in the example from UCSF, the 3' single cell RNA-Seq libraries, the primers are preloaded into the chip before single cell isolation, because you can prepare like dozens of chips with those barcoded primer in the well and store them until the day of use. So these are very stable once they are isolated in there. In the case of the UCSF study, you have an individual barcode in each of the microwell. In the case of the work from the direct library preparation plus method, it's a dual indexing barcodes. So each row and each column will receive like a combination of two barcodes. And again, these are preloaded before isolation of single cells.
Okay, and how long is the CTC enrichment and cellenONE isolation?
The CTC enrichment with the biolytic system take under 1 hour, and the isolation with the cellenONE system obviously will depend on the number of CTCs that we are isolating, but typically when we process a complete sample, it's about 1 hour to 1 hour and a half.
Okay, and our final question today. Why can't people use the chromium from 10x Genomics directly on the enriched sample?
Well, so they could, but this will be very expensive for them. Actually, there is a paper published, I think, in Scientific Report in 2018, where they did just that. And basically, out of like several thousands of cells that were sequenced, they only found 1 or 2 CTCs. And when you know the cost of running Chromium 10x protocol, it's over 10,000 euro just to get the transcriptome of 1 CTCs. It's not really efficient. So very low recovery rate, low capture rate of single CTCs, and very expensive. So we don't think that this is really implementable in the hospital as a routine test.
Okay, thank you, Guilhem. Thank you very much for your questions. And thanks once again to Guilhem for this excellent presentation. And so our 7th annual Single Cell Analysis Congress will be taking place on the 7th to the 8th of November in London in the UK as part of our SynGen series. If you would like further information on the Congress or would like a copy of the conference program, please contact the marketing department or visit the conference website. On behalf of Oxford Global, I would like to thank everybody for joining us today. I would like to say a big thank you once again to Guilhem, who has put a lot of time into making sure that this webinar was as interesting and valuable as possible. So all that remains to be said is have a great day, and we look forward to welcoming you to one of our future events. Thank you.